Hello friends, now today we are going to discuss about enzyme linked immunosorbent assay part 2, and here we are going to discuss sandwich ELISA. The sandwich ELISA, which quantifies antigens between the two layers of antibodies, that is a capture antibody and second one is a detection antibody. So what is the meaning of capture antibody? The antibody which is adsorbed on a plastic surface of well, and second one is the detection antibody which is used for the detection of antigen. So here the antigen which is sandwiched between the two antibodies. The sandwich assay uses two different antibodies that are reactive with the different epitopes on the antigen with the concentration that needs to be determined. So here we are using two different antibody. One is a capture antibody that will react with the one epitope of antigen, and second one is a detection antibody that will react with a different epitope of antigen. So this mechanism we are using for the detection of antigen. So what is the requirement for the ELISA? Here we are using solid phase support. Here this is 96 microtiter well or polystyrene beads or tubes. The antibodies are adsorbed on the plastic surface. So here you can see this is a plastic plate on which the wells are constructed, and this well we are using for the ELISA purpose. So here you can see the plastic plate which is a transparent, and here there are the 96 wells. You can see very well here, and it is a transparent. So this well we are using for the ELISA purpose. Now what is the another requirement? So here this is a enzyme conjugate. With your ELISA kit there is some bottle is available or this tube is available that contains enzyme conjugate. So what is the enzyme conjugate? We will discuss here. So here you can see the antibody which is linked with the enzyme. So how it is constructed, let's see. So here what is the enzyme conjugate or conjugated secondary antibody? Means the enzyme linked to the specific antibody. So here you can see this is the antibody which is linked to the biotin, and here you can see a long spacer. Second thing, this is a avidin or streptavidin. This is a protein which is linked with the biotin. So it makes a strong bond, and here you can see the enzymes like horseradish peroxidase, or it may be alkaline phosphatase, it may be peroxidase, it may be beta galactosidase, or it may be amylase. So here what is the substrate for this ELISA? So substrate may be tetramethylbenzidine. So here this is a colorless compound. It get converted into the colorful compound by the action of enzyme horseradish peroxidase. So it is a commonly used substrate for the ELISA. Now what is a washing solution? So it is always a phosphate buffered solution or saline, and it is a balanced salt solution which is commonly used for the as a buffer for washing. So it is very important for the washing. If it is a pure, your ELISA will be correct. Now what is a stop solution? To stop the activity of enzyme on the substrate, there is a requirement of solution that stops the activity. So the stop solution, it is a solution contains 0.16 molar sulfuric acid, which is used to terminate the activity of peroxidase on TMB reaction for the ELISA, or it is used for the termination of activity of any other enzyme or particular substrate we are using for the ELISA activity. So here the stop solution, it contains sulfuric acid, and this is responsible for the precipitation of that particular enzyme. So here the activity of enzyme, it will stop there. Now here you can see the kit. That kit contains your plastic well, the standards, and here the different types of solution which are required for the ELISA activity. Now here you can see the ELISA washer. This ELISA washer is responsible for removing unwanted things. So if it works properly, your ELISA will be accurate. Now here this is a ELISA reader. ELISA reader, it is a specialized spectrophotometer which is designed to read results of the ELISA test. Now the ELISA plate contains 96 wells. Here you can see these 96 wells are there, and these 96 wells on which we are going to apply sample, standard, blank, control. So here you can introduce your sample. Now this is a well. Now we are going to zoom this well. After zooming, it will be like this. Let's see. Now normally what you see, this is a clear well. There is nothing inside this well. But actually there is a antibody which is provided by the manufacturer. So there may be, suppose you are doing estimation of hormone, for example TSH, and TSH, it is your antigen. So the antibody for the TSH which is already adsorbed on the plastic surface of well. So suppose there is an antigen you want to detect. For the antibodies for the particular antigen which is provided by the manufacturer and which is adsorbed on the plastic surface. Now a specific captured antibody which is immobilized on a high protein binding plates by overnight incubation, and this work is done by the manufacturer and that is provided in a kit. Now the space you can see here, the space which is blocked with irrelevant protein, that is a albumin. Now we are going to add sample. Now the sample is added here. Now here you can see the serum sample, it contains antigen. So here we have to incubate it for 60 minutes. So this incubation, it varies according to the type of test which is provided by the or prepared by the manufacturer. So it varies according to kit. So here during incubation period, this antigen and antibody binds and they form a antigen antibody complex. And now here you can see the unwanted things. The unwanted antigens are there and we don't want to interfere with our reaction. So here we have to wash it. So the washing step is very important here so that we can remove unwanted thing. Now after washing you can see the steps, like after washing, here the antigen and antibody, they are linked together, and here you can see a clear well. Now here we are going to add conjugated secondary antibody which we have discussed earlier, means that is the enzyme linked to the specific antibody, and after that we are going to add specific substrate, that is a tetramethylbenzidine, and here this antibody which is linked with the enzyme, that is a horseradish peroxidase or alkaline phosphatase. Now here we are going to add enzyme conjugate. So enzyme conjugate which is added here now. Now this enzyme conjugate contains conjugated antibodies, and this antibody now they are binding with the antigen. So here you can see antigen antibody complex again. Now here this enzyme conjugate which contains unwanted excess of conjugated enzyme, and we are not interested in these antibodies, so here we have to wash it. So wash with a washer. Now after washing you can see this picture, here you can see a clear well. Now we have to zoom this. After zooming and before that we will add our substrate, that is tetramethylbenzidine. Now this is a picture after zooming, and here you can see we have added substrate. Now substrate on which the enzyme horseradish peroxidase will act, and we will get a colorful product. Now here you can see that the antigen antibodies are linked together. Now this second antibody, which contains enzyme which acts on a substrate that produce n number of products, and here if we have more antigen, there will be more binding of the secondary antibody and the more conversion of substrate into product, and here we will get more color. So this estimation can be qualitative or it can be quantitative. Okay. Now let's see the summary. This is a well which contains antibody on which we have added sample. This is our antigen which is present in sample. This is a secondary antibody with enzyme. Then this is a substrate on which the enzyme acts, and here we are getting product, and the more the product formation, there will be more color. So this is the principle of sandwich ELISA. Now here you can see the well, and the well in which we have added sample. So after the end of procedure, what happens? Let's see. Now at the end we will add stop solution, that is a 0.16 molar H2SO4, which is responsible for stopping this reaction. So if we have more color, it means that the given antigen which is present in a given sample. So it may be a qualitative, it may be a quantitative estimation. So what is the advantage of ELISA? It has high specificity since two antibodies are used and the antigen or analyte is specifically captured and detected. So it is suitable for the complex sample as antigen does not require purification prior to measurement. So this is the advantage of sandwich ELISA, and we can easily use this technique. And here you can see a color. The color can also be observed by a naked eye. So this is a good and easy method for the detection of antigen. So it's all about sandwich ELISA. Then next topic will be direct ELISA. Okay, we will discuss in a next lecture. So here this was all about sandwich ELISA. If any question you want to ask, you can write in a comment box, and please like, share and subscribe. Thank you.